everything, we have to pick and choose out of the chapters that we have what we are going to be able to do. Now, chapter 12 sets up a lot of how the nervous tissue, what are the cells, what do the cells look like, what are their special features, what are they communicating with, how are they communicating, and so there's a lot in chapter 12. So this is one of the chapters you have to dig in, okay? And it starts with kind of just organization of the nervous system. And so when we look at the organization of the nervous system, you know that there's the brain and spinal cord anatomically that are encased in bone. That is the central nervous system. And then you have the cranial nerves that have information from neurons going out, and then they get information from sensory neurons going in. And then you have your peripheral nerves, which, like cranial, are going to have some neurons that go out and talk to glands, talk to, muscles, and then you'll have sensory information coming in. Okay, so anatomically speaking, you can divide the nervous system into central nervous system, peripheral nervous system. Okay, then when you think about it, information is either coming out of the brain or coming into the brain. So when we think about that, we know that information going out is normally going to be motor, or it's going to be like glands telling your sweat glands or your salivary glands to secrete things. So motor and secretions go out, and then things that come in are going to be sensory. Okay, and the terms we sometimes call things for when it's going out versus when it's going in. If it's exiting, we use the big E for exit. So efferent means it's going out. Afferent, the A with the beginning of the word, is going to be things are coming in. Okay. So you are going to have everything needs to get to the brain in certain areas of the brain, and we're going to talk about those certain areas, and things are going to need to leave. And again, they're going to originate in certain areas, certain parts of the brain. Typically, when things go out, it's going to exit. It's part of the efferent system. When things are coming in, it's going to come afferent and enter the brain. All right. And your cranial nerves are going to carry things leaving the brain and going to most of the coming off the face, the neck, and a few areas outside of the neck and the head. Your peripheral nerves are going to come out the spinal cord, and they are going to be, again, a bunch of neurons. Some of those neurons are bringing information in, and some of those neurons are bringing information out. All right. And so things that are bringing information in, we will call them motor if they're going to skeletal muscle under voluntary control. So these are those neurons releasing acetylcholine at your neuromuscular junction. They are part of the somatic motor system. All right. If they're going to the heart, smooth muscle glands like your sweat glands or your salivary glands, all right, then they are going to be part of the autonomic nervous system. And that system is not under your control, but that system is going to have your ramping up or you're ramping down. So it's not like on off, like the skeletal muscle, you either contract the muscle or you relax. And then the organ kind of manipulates like these 5 muscles contract, these 5 muscles contract, these, and that holds tension. In your system of autonomics, you are always potentially with some of your smooth muscle, they're contracted. And you ask them to contract more or you ask them to contract less. When you contract more, you constrict things. When you contract less, you dilate things. Okay, so that's kind of how the organization goes. Now, the system has some fail safes built in, and the fail safes that are built in are called your kind of spinal reflexes. Okay, and the goal is that you are getting some sensory information that is so intense, so big that it could potentially lead to catastrophic damage before your brain was to get the information, process it, become aware of it, and then decide to do something with it. So your sensory or your spinal reflexes are there to kind of prevent that from happening. Okay. Your spinal reflexes also start to drive things like peeing and pooping. They are going to control the internal sphincters. And so when you push on those internal sphincters and you start to tell those sphincters like the bladder's full or the rectum is packed, they're going to take that information to the spinal cord. The spinal cord is going to quickly send information back to tell those areas to relax. And then your external sphincters, controlled by your somatic motor system, by your voluntary muscle, is going to then be holding it until you want to let it release. Okay, so there are lots of spinal reflexes, all right? And your book is going to go through. The simplest spinal reflex is the one at the knee where you like bang on the patella ligament. And because the patella ligament is going to be deformed and that potentially could damage the patella, damage the ligament, your information is going to carry to your spinal cord and quickly jump to muscle contractions that are going to cause your knee to extend, and by it extending, those quadriceps are going to contract. The goal would be to prevent any damage to the patella and to the bone and to the patella ligament. So the whole point is that reflex is protective, all right? That reflex is going to have a little bit different from most of our reflexes and our spinal reflexes in that it's going to have a sensory neuron that comes in. It's going to directly communicate to the motor that is going to go out and tell the quads to contract. Okay, so you have that little thong, it bumps on your patella ligament. That is going to bring information in. It's going to quickly jump to the motor pathway and send information out. So that is one of the simplest reflexes we know, and we know that it's two neurons. It's one sensory neuron, one motor neuron. They directly are in contact with each other. All of our other reflexes, okay? So all of our other reflexes are going to follow this general little schematic here. And this general little schematic is going to tell you that it's typically going to be 3 neurons in a spinal reflex, okay? There can be more, all right? But generally, we're going to have 3 neurons. We're going to have that sensory neuron that is tied to pain, temperature, some type of pressure, deformation, some type of sensory signal. And when that sensory signal, such as a hot plate or cold ice or whatever, temperature or a cut or nails or whatever, hits that skin and starts to make cells explode and starts to cause damage to those cells, these sensory neurons sitting in that connection to those sensory receptors or sitting in that area with their free nerve endings sensitive to the interstitial fluid and the chemical makeup of that fluid are going to get triggered. And they're going to, again, start an action potential that's going to travel all the way to the synaptic knob of the axon. And that axon is going to release neurotransmitter at that synapse. Okay? So that is our first neuron. It's our sensory afferent neuron bringing the information to the spinal cord. All right. At the synapse, there could be multiple neurons. Okay. One neuron might go to the brain, and he's eventually going to tell the brain, hey, you're in pain, you're hot, you're cold, damage has happened. All right. The important thing with the spinal reflex is there is going to be what's known as an interneuron. It's over here, and it is going to be our second neuron in our pathway here that is going to take the information from sensory, generate its action potential, and lead to a neurotransmitter release that then is going to come out. And if it's going to try to do skeletal muscle, it's going to be one somatic motor neuron that then goes and releases acetylcholine on our skeletal muscles and makes them contract. If it's going to need to tell the pathway of your autonomic nervous system, the autonomic nervous system takes 2 neurons to have their effect. So that's why it's showing neuron number 1, neuron number 2. Okay, but in general, most of our spinal reflexes, there's one peripheral neuron coming in, an interneuron that connects that peripheral neuron to the muscles, to the area for effect, for escape, for handling that we need to handle. And it's going to be that usual skeletal muscle somatic neuron that leaves. That's going to be in our effector. All right. For some of our more complex spinal reflexes, so if I do a cut on my hand and or I touch a hot plate, I touch it, I pull away, it might be that I have just the effectors, just the muscles contracting in that one arm. If I tried to step on something hot and I tried to pick the foot up, that's going to usually involve spinal reflexes that trigger my leg that is stepping on the hot coals to come up, but it's also going to need another interneuron to talk to the other side of my body, my left leg, and try to get me to put my weight on that leg. So again, I might have another interneuron that comes back to motor neurons on the same side as the effector or the opposite side as the effector, and trying to, balance out that if I pick this leg up, that I put my weight on the other leg. Okay. So how are you going to get asked questions? All right. You will get asked questions about the spinal pathway, as typically it's a sensory stimuli, an afferent sensory neuron, an interneuron, and then the effector. All right. And the effector, if it's going to skeletal muscle, is going to be one neuron. And we'll learn that in the autonomics, there's usually two neurons. One is called the preganglionic, and the other is called the postganglionic. Okay. And they are going to be the neurons that then lead to release of the neurotransmitter that eventually gets your heart rate to go faster, your blood vessels to some places like your skeletal muscles dilate, and then in other places like your stomach and your bladder and your kidneys constrict, so you can try to shunt blood to the area where we're going to need it, because we're going to need to potentially fight or run away from our environment. Okay. For the types of spinal reflexes, I don't need you to know any particulars or their names. But again, some of the names are, there's the knee extensor one, which is our simplest. It doesn't have any interneurons. And then you usually have your extensor flexion one, which is kind of like with the hand, you hit something hot, you pull away, you hit something cold, or you cut it, you pull away. And then if you have to do the one on the legs, usually that's going to be a cross extensor, because you're going to have to, as I flex away, I'm going to have to extend and put my counterweight and balance on the other side. Okay. So far, we've kind of talked about how the nervous system is organized. You need to kind of organize that in your head anatomically, brain and spinal cord, and then cranial and peripheral nerves. And then physiologically, things are going into the system, usually from sensory. So that's our afferent, and that's our sensory system coming in. Things are usually going to exit out our efferent nervous system, which is going to then bring system either to the skeletal muscle, so that's the somatic, or bring systems out like for smooth muscle and glands and sweating, and that's going to be the autonomic. All right. And the autonomic, you ramp up with sympathetics, like your fight or flight response, and you ramp down with parasympathetics. Okay. For spinal nerves, we tend to have receptor and stimuli, afferent neuron, interneuron, efferent neurons. Okay. Many interneurons can be in play. Another interneuron can start bringing information to the brain. Another interneuron can start bringing information to the skeletal muscle on the other side of the body to, again, kind of counterbalance whatever you're going to do. All right. Next topic is some of the information that we've gone from chapter 4. These are the major parts of the neuron. Okay, and so far all we've talked about is these multipolar or multipotent neurons. And in general, neurons are going to have many inputs, so many dendritic surfaces and ends, but one output or one axon. Okay, And the goal is that this is usually one-way transmission. Okay, so you are trying to send information to this neuron, and that information is going to summate or come together at this axon hillock. And if that axon hillock reaches a critical threshold, right, then you are going to send the signal to your axon termination sites and release neurotransmitter like acetylcholine or some other neurotransmitter. Okay, so that is how. There's usually not going to be any backward transmission, and then the dendrites usually do not send information backwards, right? One-way transition. Now, the area of the neurons that form a synapse are going to be the synaptic knobs and usually the dendritic surfaces. All right. And we've talked about, again, the synapse in relationship to the somatic motor neuron and the muscle. Remember the motor end plate? So the somatic motor neuron was releasing acetylcholine. Acetylcholine was traveling across that space, going to the motor end plate and hitting its receptor. So when neurons have synapses, they can synapse on other neurons, they can synapse on skeletal muscles, they can synapse on smooth muscles, and they can synapse on glands. Okay. And so because this is again one-way communication, all right, the neuron releasing neurotransmitter is going to be your presynaptic neuron. Okay, so it's the one that has an electrical event coming down that's causing vesicles to merge with its membrane, and it's releasing the contents of that vesicle, which is normally some type of small molecule that we call, because it came from a neuron, a neurotransmitter. And that small molecule is leaving the neuron and crossing the space and transmitting the signal onto the next cell. Okay, the receiving cell, all right, so this is our receiving cell, and this can be, again, a skeletal muscle, a smooth muscle, it can be a gland, or it can be another neuron. It's going to have its receptors for the neurotransmitter typically in its membrane close to that synapse, and it's going to, when, again, the molecule and the receptor merge, it's going to undergo some type of conformational change. That conformational change could be, again, ions come in or out of the cell, and that can change the electrical properties. It could be that the cell changes its amount of its receptors. It could be that it changes its sensitivity to its that's what some of our neuromodulators do. They change the sensitivity of the synapse by putting more or less receptors on the membrane so that the signal can be amplified or dampened down. Okay, and again, the space between is known as your synaptic cleft. All right, so again, we always have one-way communication. Remember that we typically want to put more transmitter in the synapse than we need. So technically you need 3 molecules to find the membrane bound receptors, but we're putting in more because some of it's going to, go the wrong way. Some of it's going to get pulled back into the cell, maybe reuptaked and saved for the next time. Some of it is going to, again, miss, and then some of it is going to hit the receptor, right? So we don't put just 3 molecules of receptor or of neurotransmitter out there. We put a lot of it to try to ensure and statistically speaking, increase the odds that we hit that membrane and make that change. Okay, so we just got finished with hormones, and we just learned that hormones, all right, come from certain type of root molecules. We learned that hormones, we have amino acids that we modify, all right, and they give us some of our hormones like epinephrine and norepinephrine and T3 and T4, all right, thyroid hormones, okay? And then we learn that we make proteins through transcription, and then translation, at the ribosome, all right? And we can make hormones that way. And then we learned that some of our hormones come from cholesterol, all right? And they were our steroid hormones. Okay. For our neurotransmitters, like some of our hormones, they need to be water loving, all right? And they need to be small. They need to be easily made, but they also potentially need to be either recycled easily or broken down, okay? And they need to be something that the neuron can quickly make and recreate or make and grab and send out. All right. So many of our neurotransmitters they are not going to be steroids, because remember, steroids and fat-based things, we're not going to need that because we need it to stay water loving in the synapse. So like many of our hormones, all right, our neurotransmitters are going to come from modifying amino acids. All right. And so many of the amino acids we're going to modify are going to be tyrosine again. Because remember, tyrosine is going to give us dopamine, and then technically norepinephrine is a neurotransmitter. So tyrosine gives us two big neurotransmitters. Okay. Some of the other ones out there, again, there's glutamate, there's GABA. Many of those are going to also come from some modified amino acids. All right. So when we think about some of the critical neurotransmitters need to be similar to hormones in some ways, because they need to be something that's small, easily made, something that can be recycled or broken down, but so they have to be water loving. They have to be able to leave the membrane of a cell, move through the water between the cells, the interstitial space, and then they need to bind to a receptor protein. Now, they don't go into the cell, they don't go across the membrane. And again, remember, the membrane is fat-ish. It's that phospholipid bilayer. And because these molecules are water loving, they can only have their effect if there is a receptor there. Like many of our hormones, that receptor and neurotransmitter binding is going to lead to changes across the membrane. And those changes across the membrane are usually going to involve ions coming in or ions coming out and changing electrical properties. Okay, now how it differs from hormones. All right, we're not going to make them from fat. They are not going to travel a far distance. They really are only going to travel a small little space in the synapse. They are not going to typically travel in the bloodstream. And many of our hormones bind to proteins and cause second messengers. And so they're using receptors that are triggering more inside the cell, not just one event, they're triggering multiple events. But the neurotransmitter might only need to change one event. It's changing the protein to open and let just one ion in or just one ion out. So it's not necessarily trying to do a lot of things all over the cell. It's just trying to change the electrical property at that membrane. Okay, so we've talked about norepinephrine. This one's going to be important in our sympathetic nervous system. We've talked about acetylcholine. Acetylcholine is our motor for skeletal muscle. So our somatic motor neurons and skeletal muscle communication. We're also going to learn that acetylcholine is important in the parasympathetic nervous system. Lots of people take serotonin or serotonin inhibitors, serotonin uptake inhibitors. Serotonin we associate with mood, and again, many people take it for anxiety, depression, and it works in certain areas of our brain associated with our emotions, with our mood. Dopamine, we know when people don't make dopamine, that's what some of the shaking with our Parkinson's is located. So again, dopamine is part of a neurotransmitter that is part of the connections to eventually get our skeletal muscles to contract and smooth out and key into those contractions being not in a shaky or jerky way. Okay, so do you have to learn what every single neurotransmitter is? No. All right. Eventually, when you get to a pharmacology class, you will learn a lot of drugs, especially when we talk about mental health and emotions and moods and attention, because those drugs are trying to make the synapse either have more neurotransmitter or less neurotransmitter in the synapse for longer, or neurotransmitter in the synapse for less time. Or they're trying to make the receptor blocked or the receptor more available and more affinity for the neurotransmitter. And you're messing with the synapse to try to make the neuron receiving the neurotransmitter up its sensitivity or down, bring its sensitivity. Okay. So again, we don't have to learn all of them, but we do know that norepinephrine and epinephrine and dopamine, they come from tyrosine. And we know norepinephrine and epinephrine as a group of hormones are known as your catecholamines. Okay. We know acetylcholine is big in your somatic motor neurons telling skeletal muscles to contract. And we know acetylcholine is also a big thing in our parasympathetic system. Okay. All right. The other thing to talk about is, again, think about morphine, think about endorphins. Neurotransmitters are what's happening between the cells, the neuron sending the signal and the neuron receiving the signal. Neuromodulators are going to be, again, other molecules that bind to other receptors that might cause more membrane bound receptors to show up on the membrane, making that cell more responsive. Or in terms of pain, taking some of these receptors and pulling them into the cell, making the number of receptors on our pain signaling pathways to be less available. And if there's less pain receptors on the membrane, then it's harder to get that signal to the pain neurons to send the pain message to your brain that, ow, I'm in pain. And that's what some of our morphine and that's what some of our, um, you know, drugs do when they're helping us dampen our pain. They're not actually changing the amount of neurotransmitter. They're changing the amount of receptor or the sensitivity of the receptor to bind to that neurotransmitter, the odds of it finding that neurotransmitter. And by doing that, you're making it less likely then for that signal to get sent to your brain. Okay. All right. We have two more topics I want to get through before we call it a day. All right. So far, in what we've looked at for neurons, we've looked at neurons that somewhat always look like they have, this little one eye for its nucleus, its cell body. It's got lots of wild hair, which is its dendritic surface. And then it has a long axon that might end in 3 or 4 or 5 little synaptic knobs. Okay. And this is, again, going to be one of the neurons that we've very well categorized. We'll have it as a model to look at in our lab. All right. And it's one that, again, it's very clear. It's the one-eyed monster. It is our multipolar neuron. All right. And it's very common because this is, again, what our somatic motor neurons look like. The head is going to, for the somatic motor neurons, it's going to sit in our spinal cord. All right. And then its axon, it's going to exit and it's going to become part of your peripheral neuron. All right. Your peripheral nerve, sorry. And then it's going to travel to the skeletal muscle it controls, and it's going to synapse at their motor end plates. And when you ask it to contract, you ask if this neuron controls 5 muscle fibers, you're asking all 5 of those muscle fibers to contract at one time. Not every neuron in the body looks like this. Okay. So if we were to go back again and look at my little spinal thing, this little neuron is drawn with this little kind of cell body coming off of it. All right. Our little multipotent one-eyed monsters are usually drawn like that. The axon is this little triangle, all right, where it ends, and the cell body and its like crazy hair is its head. Okay. But when we look at, there is another type of neuron out there, and that other type of neuron is going to have its cell body that is going to look like a little leaf that comes off of this main branch. All right. This is going to be one of our unipolar neurons. Okay. Now, the difference here is the cell body is going to typically be located somewhere off of the long kind of the axis here, the long kind of branch. That branch may have a dendritic surface of free nerve endings, or it might have some type of connection to a sensory cell or a sensory object that that is connected to being able to sense whatever's going on. Okay, so the difference here is that technically this area looks like a long movement of electrical energy to the cell body. It's not a true axon. It's like the incoming axon, and then you will have the outgoing axon that's going to depart from the cell body and then go to its synaptic knobs or its synaptic terminal ends. Okay, this is going to, again, usually be sensory neurons, all right? And it's usually tied to any of the general and some of our special receptors that we have out there, okay? So in our spinal nerve, so let's say I look at the nerve that's coming out at T1. T1 may have some sensory neurons coming in, all right? And then it may have some motor neurons going out towards my arm. And that whole spinal nerve, all right, is is a combination of some information coming in, some information coming out. And so if I was to somehow lose my T1 peripheral nerve because somehow I have a bacterial infection and it kills it, I am going to lose all the sensory information that would be coming in. So let's say T1 is like sensory information for my armpit and controlling my triceps. I would potentially lose my ability to sweat in my armpit. I would potentially lose my tricep muscle being able to counterbalance my bicep. And let's say I would lose my feeling, my touch, my pressure of any time you like try to mess with my tricep area. And maybe if you put ice or a cigarette on me, I wouldn't feel it. Why? Because I would lose all that sensory and I would lose all that motor going out. Okay. All right. When we get to certain areas of the brain, we're going to see that there are even some other type of neurons that have a feature or a place in our system. All right. One of these is known as the bipolar neuron. And the difference between it and the unipolar is the unipolar branches off. The bipolar, the cell body is directly in the center. So for this guy, all right, you have an axon, a dendritic axon bringing information to the cell body, and then you have a dendritic, or I'm sorry, the normal axon bringing information away. For this one, for the bipolar, the cell body is directly in the center, and so it's a little more defined. One is the dendritic axon, and one is the efferent axon, bringing information to your synaptic knobs. There is only one place in the human body that has bipolar neurons, and it is in your eyes. And I can show you a model later on, but these are the cells. So right here, this cell that it's connected to, this is your rod or your cone cells that are the um, the receptor cells for stimulating by light energy. And so when they are stimulated, they release neurotransmitter that then stimulates this bipolar neuron. And then this bipolar neuron takes that information to a standard kind of interneuron, All right. So this is your primary sensory neuron, then it brings it to a interneuron, so a second neuron in the pathway that then will bring the information towards you as part of your cranial nerve towards your occipital lobe, towards the back of your brain to be processed. All right. And the last type of neuron that we have, and this one's going to be in many of the cortex or the gray matter regions where we don't need the neurons to travel a little far distance. They just need to communicate to the neurons around them. You're going to have something that doesn't really look like it has a very true axon, or it's hard to tell which is the axon. And so it looks kind of like it doesn't have any extended branches, and it's without a axon. So remember, a in front of something usually means without. All right. So anaxonic neurons. So it's without an axon neuron. Okay. So these are the types of neurons that we know of. And again, most of them are going to have something that's an axon. So you can apply axolema, you can apply axoplasm, you can apply those terms when you're talking about the axon versus the cell body, the nissel bodies, the neurofilaments, again, just general terms that we refer to for some of the cytoplasm and um, the organelles inside of the neuron. Okay. Now, when we did chapter 4, we said that all supporting cells were called neuroglial cells. Now we're going to ask that you actually learn what are the individual types of neuroglial cells. Okay. And so the neuroglial cells, there are again 4 of them that are in the central nervous system. Okay. The neuron is a terminally differentiated cell. You cannot ask this neuron to go through mitosis, because to go through mitosis and try to split its DNA and split its cytoplasm and split the axon, that is a hot mess. That is tricky to do. So you have, when the neurons fully develop into their shape, they are done. You either keep them alive or you die and you lose their function. So the neuroglial cells are going to be around the neurons trying to keep them alive. The neurons are so important that we're not going to let blood, we're not going to let white blood cells, we're not going to let many of the normal things that kind of go around and check for foreign invaders. We're not going to let those things come near our neurons. All right. So our neuroglial cells are going to have to help separate the neuron from the white blood cells, from our blood, from other aspects of our body, keep them isolated. And they're also going to have to take up some of the roles of, well, somebody's going to have to help get rid of trash, get rid of gook, get rid of inappropriate things that show up in and around the neurons. Okay. So when we look at the neuroglial cells in our central nervous system, we are going to have epidermal cells, astrocytes, oligodendrocytes, and microglial cells. Now, a little bit of their name tells you maybe what they look like or what they do. So when you see something that's epi, all right, EP, epithelial, all right, you should think of, well, these cells are going to typically be making cerebral spinal fluid, and they are going to line the ventricles and the central canal where cerebral spinal fluid is going to flow. All right. So they are going to be some barrier type cells that help create an environment where only cerebral spinal fluid is allowed to be in and around our neuron. All right. Astrocytes are going to, again, have these kind of star-like shapes. Where are they? Do-do-do-do-do. All right, here's an astrocyte. Okay, and it is going to have little projections that are going to, again, where the blood and where the connective tissue and the outside tissues try to come close in contact to our neurons, they are going to try to make our blood brain barrier. And they are going to work to have wraparounds to prevent things from the blood from being able to get out. So white blood cells taking a ride in the blood won't be able to exit because the astrocytes are going to block it off, especially as it travels through areas where there's neural tissue nearby. Okay. Our oligodendrocytes, I like to think of them as an octopus cell. these are going to be the cells that are going to myelinate your neurons. Okay. And they are going to myelinate your neurons. So if they're like little octopuses, they are going to have these little arms that are going to then go and wrap around one neuron, maybe 3 to 4 or 5 times. But they also have the ability to maybe have two neurons or other neurons they wrap around, and they're able to then myelinate multiple places on one neuron or multiple neurons in a few places. Right? And remember, the whole point of myelination is when I get a signal, I jump to the nodes of Ravanier and I'm able to move from the start of the axon to the end of the axon. I'm able to move down that line faster than if I have to like go heel to toe, heel to toe, heel to toe, and walk like that. Okay. The last type of cell found again in your brain and spinal cord is going to be your microglial cell. And your microglial cell is going to take the role of kind of being the trash man. They are going to have the ability to endocytose or phagocytose material and pull out stuff that debris, pieces and parts of the neuron, maybe even excess neurotransmitter. They can help relieve and get that out of the way. Okay, so those are your neuroglial cells that you have to know by name, by shape, by role inside the central nervous system. The neurons that are going to be in the peripheral, all right, we are going to have these cells, especially in places where we're going to have lots of sensory neurons that gaggle together. They are going to just kind of sit around and protect those cells. So they are going to be our satellite cells, and we're going to find them in places like our dorsal root ganglia, in our sympathetic ganglia areas. So any place where we have one neuron synapsing with another, and we have that again where the sympathetic neurons have the first one in the pathway and the second. That is going to be again in a place where we're going to have lots of satellite cells because it needs to protect the cell body of the neuron receiving the message, and it needs to protect the axon, the terminating ends of the axon that's giving the message. Okay. The other cell in the periphery is going to go back to being our myelinating cells, and these are our Schwann cells. Now, the difference between the Schwann cell and the oligodendrocyte is the Schwann cell can only wrap around one part of one axon. Okay, and when it wraps around, it forms this area around the axon known as the neurolema. Okay, so in order to myelinate, let's say an axon in 4 different spots, you would need in the peripheral nerve, you would need 4 different Schwann cells. If you wanted the axon to be myelinated in the central nervous system, so the brain or the spinal cord, and you have these 4 different little spots, that one oligodendrocyte could potentially myelinate all 4 spots. Okay, now the advantage to that. So as it goes through your book, because we have Schwann cells that are only responsible for this one distinctive part of the axon, and then there's another Schwann cell for the next, if there is damage, the Schwann cells are known to help regrow the axon, keep its little tunnel, little space available so it can reconnect back to the muscle or back to the gland that it's supposed to connect to. And they are helping the nervous system regrow when the nervous system gets damaged. If you damage areas in the spinal cord or the brain, you don't get that regrowth. So the oligodendrocyte doesn't have the ability to do the regrowth like the Schwann cells does in the periphery. And so that's one of the reasons why if you have surgery and you're like, you have this big numb spot from your scar, 2-3 years later, your numb spot might be just the size of your finger. It's not a big circle anymore because some of those sensory neurons myelinated by Schwann cells have able to reconnect to the area they are connected to from before because the Schwann cells help them get rid of the damage and regrow and reconnect to their part of their sensory receptor. Okay, so the last thing I want to leave you with is there is a lot going on electrically. Okay, so by the end of this chapter, you should be able to talk me through, here's the extracellular fluid, here's the intracellular fluid. You should be able to tell me why sodium wants to, as I can, okay, why sodium wants to come into the cell, why potassium wants to exit the cell. You should be able to tell me why we seem to have high amounts of sodium out versus why we have high amounts of potassium inside the cell. And you should be able to tell me what is resting membrane potential for a neuron. All right, you should be able to tell me what are exciting graded potentials versus what are inhibitory grading potentials. And then you should be able to tell me what an action potential is. Okay, so there's a lot of vocabulary and a lot to go over this. There are some really good videos all over YouTube, Khan Academy, other neurophysiology people that talk you through this concept. I used to have one guy that I would make everybody watch, but his videos disappeared. So it might be that I'll try to put it at the end of the week. Again, some videos in addition to mine from YouTube to watch that might help explain a little bit better why and how and what's going on. You have to know this because one, we're going to talk about it, and you should talk about it in your 208 class because how ions flow is going to be important on how the heart knows how to contract and how we get the contraction to get faster or slower. Okay, so this is really, really important. And this is the hard part of this class because we are really in microbiology in the weeds of caring about ions across the membrane. All right, and that's hard stuff. But read through the book. I think the book is terrible, but read through the book, make your notes, watch the YouTube videos I have, make your notes, go and consider watching Wendy. I'm trying to write Wendy Riggs on YouTube, watch her videos, go and visit Khan Academy and do a little research and get your head to wrap around how do I establish resting membrane potential of the neuron? How do I excite the neuron and get an excitatory potential? How do I get an inhibitory potential? And then how do I, at the axon, when I hit threshold, how do I get an action potential and what is it that happens during the action potential? Which ions come in, which ions go out? What do we call depolarization? What do we call repolarization? What do we call hyperpolarization? Okay, and that's where I have to leave you because it is officially 2 o'clock and I have another meeting to go to and I need to go check